Welcome to the flip lecture on the structure of DNA. Now, as with all these flip lectures, it's up to you, I guess, to know your syllabus and know how much detail to extract from these videos, because not everything I say completely fits in with all syllabi everywhere, or what level you're studying at. So for the lower level science courses or junior science courses, you just take the basic information. When I say, like, oh, this is quite basic, this is, you know, the intro. And then if you're doing higher level biology, then you can take a little bit more in and add to your syllabi. So like anything, know the syllabi that you need to, and the detail that you need to get from the videos. And of course, if you just have a love of biology, then you just absorb it all. Alright, so let's look at the structure of DNA. And as you can see here with the model, the structure here is a helical structure. Helix means to coil around. And you can see it's like a staircase. And we can also see that there are backbones. So we can have a backbone here, sugar phosphate backbone, and we have a complementary backbone. Okay, so the two twist around each other, like so. So we have a backbone, sugar phosphate backbone. Now, what is that sugar, I hear you ask? Well, I'm about to tell you. So let me draw the sugar for you. Okay, that's the sugar. Now this particular sugar has got 5 carbons, and so we name that 1, 2, 3, 4, and 5. So that's the structure of the ribose, the deoxyribose. Now I'm saying deoxy because there's no oxygen here. If this was just a normal ribose sugar, it would have an oxygen there. It doesn't have an oxygen there in DNA, and so therefore it's called deoxyribose. There we go, just add that there. A little bit smudged, but hey, that's the way the universe works. Alright, so that's our deoxyribose sugar. And the name of DNA is deoxyribo. Right, so there's our sugar portion. And then we have nucleic acid. And so the nucleic acid comes from the nitrogenous bases. And there's 2 types of bases. You can see here in the middle, the steps are the bases. And there's 2 types of nitrogenous bases, okay? In higher biology, you start talking about pyrimidines, etc., but we're not going to worry about that, but we're just going to know that they are ringed structures, okay, the ring structures. And those ring structures have different names. And we have the different colours here. And a good way to remember it is, and I'm running out of a bit of room here, but we'll just extend over here. So we got G, C, A, and T, and we can remember that by good chromosomes always twist. Okay, so the G and the T combine each other, A and the T combine each other. And they do so by using hydrogen bonds. So the hydrogen bonds connect them together. We'll draw that up soon. So that's the basic structure of DNA. Helical structure, backbone sugar phosphate, the deoxyribose sugar, and then we have the steps and nitrogenous bases. So I'll draw it up a little bit in the next section, and we'll talk about that. And we're back to the next section here. So what we're going to be looking at is the strands and how they're arranged. So at a very basic level, this is sugar, phosphate, sugar, phosphate, sugar. And coming off the sugar is a base. And what we have to realize, though, is one strand is actually upside down compared to the other strand. So remember we had said that in these sugars, we can name the carbons. We said it was a 5-carbon sugar. So this was oxygen, if you remember, because there it is over there. So this is 1, 2, 3, 4, but then the 5th one was up here in the bond here, right? It's not shown in this basic diagram. So we've got 1, 2, 3, 4, 5. And the phosphate joins to the 5th carbon. And then we bond via a 3rd carbon down here, right? So in more detail over here, you can see that we've got 1, 2, 3, 4, 5. And so what's happening is the 5th carbon is bonding with the phosphate group. So here's the phosphate group. and we're getting a bond forming here, and then the third carbon between the hydroxide group here and the hydroxide group there, they will bond by removal of a water molecule. Okay, to those who may study chemistry at a high level will know that's a condensation reaction. So those two combine to give you this bond here. Okay, and so this is bonding from 5, 3, 5, 3. And so we can see here, in general, we call this the 5' end, and we call this the 3' end. Now, if I was to make that again and put it here, then the bases wouldn't be facing each other. So in order for the bases to face each other, we need to turn one upside down, and then it can come in on that direction. So let me just rub this section out here and redraw it. Sorry about the noise. I'll redraw it and be back in a sec. Alright, it's a weird and wacky drawing there. It's pretty hard to draw upside down and then back to front, so I hope everything works out. If not, well, you know what I'm doing anyway. We've got the phosphate group and the sugar group. This has been turned upside down so that it can fit together. And if that was the 5', then now this is the 5' end, and that's the 3'. Okay, so with a little bit more detail, we've drawn it as a basic structure here, but we've stepped it up a little bit in talking about the direction of the chains, which is very, very important when you do more sophisticated biology and start talking about copying DNA, when the enzymes move from the 5' to the 3' and all that sort of stuff, which is in later videos. This understanding of what that means is very, very important. So we can see here that we've got a ladder structure. We've got our phosphate backbones, and these bases here will bond via hydrogen bonds. Okay, and the hydrogen bonds hold those two together. So that's the basic structure of DNA. Hopefully that makes sense, and I'll see you in another video when we get a little bit more structure and talk about genes, chromosomes, etc.